Mohammed Umar Farooq biological video. Please make sure to subscribe, like and share. Ask the question for answer and comment about the artwork. You can also answer question to each other for solving the problem. So here we will discuss about the cell cycle. We will just understand the cell cycle. And next video we will also understand the mitosis. So the first, let's begin to understand the cell cycle. So the period from the time a cell is produced until it completes mitosis is called cell cycle. For example, the cell division. So the complete process it's known as cell cycle. So this cell cycle is again and again to produce the copy of the cell. So that's it's important, that is the meaning for the mitosis. So the cell division of the somatic cell, which they are used for the growth and development. So the division of nucleus is the mitosis, while the cytokinesis is the division of the cytoplasm. So it means the karyokinesis is also known as the mitosis, while the cytokinesis it's mean the division complete. While that is all thing is occur in the interphase. So this interphase is known as the cell cycle. So the cell grows and carries out its various metabolic process during interphase. So the interphase is important for the checkpoint and correction of the DNA replication and correction of the protein synthesis for the precursor of division. So here is I want to draw the simple diagram to understand the cell cycle. So the cell cycle is here, which that is first of all the growth one phase. So this growth one phase is RNA and functional protein synthesis. So this functional protein synthesis is the growth one, while the synthesis phase, which that is known as the S phase, is also DNA replication. So the DNA will be double from 2n diploid to tetraploid. For example, the triplet, basically double of the DNA. So that means the 4n. Next we will discuss also. But here the structure protein synthesis will also occur during the growth 2 phase. So this is the G2 phase. So the G2 phase contains a structural protein synthesis. So that is mean the mitosis and cytokinesis that will be occur after the G2 phase. So that is all thing is due to the checkpoint. So the checkpoint is used for the correction of the cell division. So here is I am drawing the cell, which that is normally cell, and this cell will be divided into two cells. So the whole process in this division we will discuss in this video by understanding the cell cycle. So the cell cycle means the cycle will be complete to again enter into the interphase. So here's the G1 phase. So the G1 phase is basically contain basically 2n cell after the mitosis that will be reproduced, and this G1 phase contain a single cell, for example, which that is want to ready to interphase. So when go into the interphase, and here is that there are many chemical processes as well as that their checkpoint will be occur. So here we will discuss with the detail. So here the 2n is contain a cell which that going into the interphase after the M phase or mitosis phase or mitotic division. So that's G1 phase entering, and after this is interphase, which that is a different process we will discuss also. So here is the interphase. After the interphase completion, for example G1 phase with the middle is known as the restriction point or G1 checkpoint. So this G1 checkpoint is important for restriction point or cell genome size checking or something. So this G1 checkpoint is contain nutrients, which that is either nutrients is deficient or efficient, or growth factor also responsible for this checkpoint, and DNA damage checking. After this DNA damage checking, the G0 basically G0 phase will be occur is known as a growth factor, which that is differentiation of the cell as into convert specialized. So this specialization of cell is the undividable. We will discuss later. So first of all, next here is the G1 phase will be occur. So here this growth factor is also used. So the 2n chromosome will remain in this phase, while the next will be synthesis phase. So the meaning of synthesis phase, the synthesis of DNA. So the meaning of synthesis of DNA means the DNA replication. The DNA replication means the double of the DNA. So the DNA synthesis and synthesis of organelle also. So the double of the organelle for the separation of into two cells. So here is the DNA will be double. So organelle will be double, and DNA synthesis, DNA replication, it means the double of the DNA. So in the cell, the DNA will be double mean the 4n. So the conversion of chromosome into 2n into 4n. For example, the double DNA, that is the daughter DNA from the parental DNA, which that convert. So that means this is the number of chromosome from one pair is come from the mother, and one is come from the father. So that will be become of double. So that will be next phase is the G2 phase. So the G2 phase is we will discuss. So the G2 growth, basically growth and preparation for mitosis. So this is the preparation, and G2 phase is crucial to process for cell division. So that is important. So the G2 phase is a different metabolic activity we will discuss also. So this is the basically go. So while that means the cell growth. So that means the G2 phase is used for the cell growth, preparation for mitosis. So the G2 phase is crucial to process for cell division. So the ready to cell division, and they are nutrient dense. So here is a 4n, which that is go into the mitotic phase. So the cell growth. So here is the 4n chromosome are double of the DNA, which that undergo into the mitosis with the different phases. So the prophase, metaphase, anaphase, telophase, and cytokinesis. So that prophase mean the DNA will be double also, but metaphase is going to the metaplate of the cell, which that allow the chromosome lengthwise inside middle of the cell. So the anaphase is basically anaphase promoting complex is used to give the anaphase. So this anaphase is used for the separation of the 4n into 2n, but remain inside of the cell and not divide. But the telophase is used for the separation completely in the corner of the cell, and this corner of the cell the chromosome will be divided, while the cytokinesis mean the cytoplasm will be first of all the karyokinesis after the late telophase to produce the two nucleus, while this two nucleus is converted into the cytokinesis by the restriction pinch inside of the cytoplasm to divide. So that means the actin and myosin are present, which that is used for the contraction of the cytoplasm for the separation. So the anaphase promoting complex is also used for the separation. So that means the anaphase time, the separation of the chromosome. Alright. So next here we will discuss about the checkpoint and the process of the controlling of the cell cycle. Next video we will also understand the uncontrolled cell division as a cancerous cell. So that is the different video, but here we will discuss. So here is a G1 growth 1 phase CD cyclin dependent kinase enzyme are present. So this is the cyclin dependent, which that depend to the cyclin. So that is present. So while here is the mitotic cyclic dependent kinase enzyme is also present, and there are many kinases enzyme are present, but here we will discuss about the just for understanding. So here is the mitotic CDK are present, which that go into the mitotic cell division beginning. So that means here is the CDK mitotic division, which that's bind with the cyclin is due to the cyclin dependent kinase. So the kinase enzyme used to cyclin bind by the using of ATP to convert ADP, and that's phosphate group is used as a helper binding factor. So that's active mitotic CDK cyclin will be activate. So this activated maturation promoting factor MPF is a factor which that is give the maturation of this complex. So this complex will be mature, and after this will be triggered to the mitotic division. So the mitotic phase will be triggered by this activity. So this is the checkpoint which that check and stimulate the mitotic division, and that's mitotic phase, M phase. After all, where the anaphase will begin to separation. So that is the anaphase promoting complex will trigger, that is the triggering mechanism to separate the chromosome. So here is the beginning of the mitotic phase, that is the G2 checkpoint before the going into the mitotic phase. So the cell size and DNA replication will be checked. Where the DNA will be damaged, that will be repaired before the going into the mitotic phase. So after all, that will be cell go into the mitotic phase for the division into two daughter cell. So right, I hope you make sense about this concept. But here we will also discuss about this metaphase checkpoint. So the metaphase checkpoint, which that is used for the growth factor, there are many growth factor which that is used for metaphase checkpoint. So the metaphase mean the chromosome will be lengthwise are present between the cell. So the chromosome spindle attachment. So the attachment of the spindle into the centromere of the chromosome is occurred by this metaphase checkpoint. So the growth factor is used for it, while the commitment to divide and the control point 2, which are used for the commitment to divide. So after all, that's metaphase will begin to go into the anaphase, and anaphase is used for the division. So that means the chromosome will be separate and the centromere will be separate. So that is chromatin is completely separated from the um, four head of the chromatin convert into the two head of the chromatin, half. So that means again will be 2n will be produced. I hope you make sense about this. So here is the spindle attached due to the metaphase checkpoint. So the metaphase checkpoint anaphase will begin, and this anaphase promoting complex will be formed, and this used for the trigger to go into the telophase and cytokinesis, as well as the karyokinesis. Karyo means cell nuclear division, while the cytokinesis means the cytoplasm division. So after all, these active mitotic CDK and cyclin complex will be degenerate. So they're destroyed by inactivase enzyme. So this inactivase enzyme destroying, and this destroying of the inactivase enzyme to suppress. So that next mitotic CDK will be used for the next cycle. So here is the G1 CDK cyclin dependent kinase G1, which that is also go and bind with the G1 cyclin. So the binding of the G1 cyclin with the G1 CDK cyclin active. So this cyclin activation. Now here G1 cyclin will attach with us G1 CDK, so to become a activation by the using of the kinases enzyme. So that means here is the restriction point will be occur. So this restriction point is the G1 checkpoint. So the ATP use again the phosphate group, which that is donation. So here is the basically G1 CDK is cyclin active will be formed. This complex will begin to stimulate the restriction point, G1 checkpoint. After the G1 checkpoint, so the growth factor is used, as well as here is the restriction point. Cell genome size will be checked and the size of the cell will be checked, and G1 checkpoint is made up of nutrients, using checkpoint of the nutrients and checkpoint of the growth factor, as well as the checkpoint of the DNA damage. Where the DNA damage will be occur, that's correction will begin. So here is the DNA which that is checked, where the DNA methylation and acetylation will be occur, as well as the DNA fragment will be lost. So these fragment will be repaired again. So here the G0 phase will be entered, that is the resting state. For the requirement of the cell, specialization of the cell, for example muscle tissue, adipose tissue or nervous tissue, and more the liver is contain a hepatocyte, which that is also divide, but that is go into the G0 phase, but that is go into the semi G0 phase, which that is a growth 0 phase, and that is also divide itself, the liver cell, within the G0 phase, due to the different processes for the detoxification, and there are many metabolic effect in the liver. So that's why important thing is that the hepatocyte is also dividable. So the specialization of cell is undividable, while the liver is a different as compared to T muscle cell, adipose cell, as well as nervous tissue or nerve cell, or osteocyte or something, or hemoglobin, etc., etc., or white blood cell and leukocyte or platelets are different things. So that's after these things, the degraded G1 cyclin. So the G1 cyclin will be degraded and the G1 CDK will be left behind, and that's go again for the preparation in the G1 phase in the next cycle. So right. So the mitotic CDK is also circulate, and that's again activated in the next phase of the mitotic division. So here is the G1 cyclin, and here it basically that is the active mitotic CDK will degenerate and go again into the cycle. So here is the M phase, which that is used for the metaphase checkpoint. So here is the metaphase checkpoint, and after the degeneration and again attachment of the G1 cyclin, and again this G1 cyclin is a G1 CDK cyclin active, and that means degenerate again, and this G1 cyclin will be degenerate, and that's go into G1 CDK again enter into the next cycle for preparation. So here the mitotic CDK is also follow this cycle. So that's mitotic CDK will be go into again for the using by the binding of cyclin due to the ATP usage. So here is you can see that basically here is the mitotic division will be occur. Anyhow, now here we will discuss just basically here is you can say that 2n convert into the 4n before the mitotic division, and after the mitotic division complete, that's 2n again will be convert, that is haploid. So that's all thing is done here. I hope you make sense about the cell cycle, and next video we will also understand about the mitosis. So that is very easy and easy to understand this mitotic division. But anyhow, I will make a video in the next video. So